Today, we're going to talk about stochastic extinction for viral infection under the influence of defective interfering particles. So, you may have seen a picture like this before. This is the Ebola virus infecting a cell. And while you may be comfortable with this idea of viral infection, you may not necessarily know what defective interfering particles are or what that means with relation to this topic. So, let's ask the question, what are defective interfering particles? Defective interfering particles, or DIPs, as we'll call them from here on out, naturally occur during RNA viral infections. They are virus-like particles that happen when there are deletions in the genomes of the normal virus during RNA replication. And what this means is that, as can be seen on the right, while the normal virus can reproduce within a cell, the DIPs cannot. However, if the DIPs and the normal virus are co-infecting a cell, then the DIPs can reproduce because they'll steal the resources from the normal virus and make more of themselves. But the significant thing about this is that if they outcompete the normal virus, then the infection in the cell stops because the normal virus is no longer there and the DIPs are left alone and can't reproduce. This raises an important biomedical question, which is, can one devise a therapeutic strategy to stop viral infection using DIPs? To answer this question, we first looked back at a deterministic model that was created around this concept. It was based off of a chemical reaction network seen on the top that depicts this co-infection. And based off of this chemical reaction network, a system of ODE, or ordinary differential equations, was then developed, which is seen on the bottom. The significant thing about this is that they can then be plotted, as can be seen on the right here. We can assess the dynamics of this interaction. However, this does not account for the randomness and extinction that you might see in an actual real-world infection. In order to fix this problem, we came up with a stochastic equation and plotted it along with this deterministic equation and model. This can be seen in this figure on the right. As can be seen, while the dashed lines, which depict the deterministic model, only approach zero, the stochastic model actually hits zero and dies off. And it is much more random, as can be seen by the zigzag nature of the curve. This more accurately depicts what will be happening in the real world during infection. And now that we have this stochastic model, we can then better assess the extinction probabilities. All of these extinction probabilities are dependent on these other conditions, the initial conditions, the parameters, and the noise level of the stochastic model. So using all these things together, we can then move forward and start looking at the extinction probabilities and asking, how do we solve and look further into this important question? I'd like to thank Dr. Louis Fan and Johnny Yang at Indiana University for their help on this project, as well as the Center for Excellence for Women and Technology at IU.